Hello.
Hi.
Hi, how are you?
I'm on that CompuCell 3D course, and that's what I did last week, besides buy a new camera.
Yeah. Well, that's great. How was the course?
Very intense. I don't know if it's very useful to me because there's not enough link with forces, exact forces and distances. It does do code and show cell behavior. It does that fairly well, but if you have to do something that's physical, I don't know.
So much. Yeah.
So yeah, I could show it to you.
Yeah.
Actually, their Boolean part of the course would work for your most recent paper. And you could take your paper and you could do the Boolean part of CompuCell 3D on your paper, and it would make a very interesting addition.
I don't know. Yeah, I mean, you have to think about some of the things because this paper is so big and it has like a lot of things that are sort of there and they could be followed up on. So I have to think about that.
Anyway, CompuCell 3D is accessible, and I have a bunch of intro videos. They're on YouTube.
Okay. So. Yeah. Let's see. I actually have the web page. It's very unapologetically 1990s design here. But this is the summer school. I guess they're having a hackathon this weekend, and so they have like the different links here. So you have the YouTube videos, I guess from 2023. They'll probably post for 2024.
Yes, that's I'm involved in that because I proposed fixed model where you have a line of cells, and if you perturb the first one and it turns into a columnar cell, how does that affect its neighbor or neighbors?
Yeah.
And how does that go down the line? So I've got a model for that. They've looked at somite division with CompuCell 3D. And I said, had they considered Rayleigh instability? Like, I really wanted to know where they had incorporated that. And they knew about Rayleigh instability, so I'm relieved, but I still don't know how they managed to incorporate that into the model, or if they did.
Yeah.
I'm out of sync. So I'm going to show off my picture because I'm out of sync.
Oh, okay. All right. Yeah. So this is this kind of goes over some of the details of the what was covered in the course. So Monday through I guess Sunday. That seems like a lot.
Yes. It was. I went to their Python boot camp on Sunday, so I went for 8 days, and I was basically exhausted.
Yeah, I imagine.
Yeah, hard to think.
Yeah. So the Python boot camp was before the course, so you could get a crash course in Python.
Yes, it's very, very C-like. Like I've certainly seen that before, and if I wanted to program in Python, I should just kind of take a course in it, and then I'd know all of the syntax foibles for it. And then I'd be able to do it. Yeah. Anyway, the whole thing's exhausting.
Yeah. Yeah, it looks like there is a lot of stuff here. You had the introduction to the platform, virtual tissue modeling introduction, basics and intro to cellular POTS modeling, which of course is kind of like a cellular automata, but it's It's a physics model, but it doesn't model mechanics necessarily. And then the next day you had fundamentals of the modeling process, so properties and constraints, cell growth and division, then chemical gradients on Wednesday, working with Python API, building a tumor model, then forces on cells, chemotaxis, cell layouts. So they're kind of working up with concepts, working up with different ways to implement stuff in the platform. I see Boolean network modeling. Yeah, that was on Friday. So yeah, Boolean networks are interesting and useful. Something we haven't explored a lot in the group, but it's probably is useful for especially like gene regulatory network modeling and things like that.
That looked very interesting. The Boolean network was for, especially like I said, for your paper.
Yeah. So then there was, of course, compartmental cells, focal point plasticity, data wrangling, best practices for parameterization. So I guess these modules were different from these modules. Then vector fields, planar cell polarity, tissue folding, and then they were prepping for the hackathon, which of course is probably just implementing some of these things more.
I don't know if it's possible for me to do anything. Later on, if you want me to present what I did for a lightning talk, I could do that.
Okay, that sounds good. Do you want to do that later? Or?
Later, yeah.
All right.
Maybe the noise will have calmed down. You never know.
Okay.
It's a zoo here.
Right.
4 dogs.
Yeah.
4. Hello.
All right. Well, I'll come back to you later. We'll see.
Okay.
All right. So thank you, Susan, for kind of talking about that and going over that with us. Yeah, that looks like it was a full week, and of course you can catch up on YouTube. And there are a lot of concepts that we could find useful for our work. The paper that we've recently put out on differentiation waves. There are a lot of other approaches we could use. Boolean networks being one, but there are other things that we'd like to do going forward. I mean, last figure in the paper is kind of features this model of the Drosophila imaginal disk, and it has like this wave going across it. And being able to model that more explicitly with gene expression and with the morphological changes would be kind of interesting. I don't know how much of that you could model in CompuCell 3D the way we would like to model it, but that would be something to talk about for another paper, say. So, hello, Jesse and Pakhi. How are you? Hello. Yeah.
I'm much. I finally got some good sleep, and I'm ready to push forward on some things again. It was a very difficult weekend, to be honest. But I'm looking forward to things here. I don't think Lucas is here. I was going to ask a little more about some of that Lucas project and see if for ways that I can also help with at least using Drupal to facilitate some DevoWorm stuff too, or like help advertise things, or if you want to do stuff for anything. I know we had this conversation with Aurel. We had a big conversation for Aurel in the future and fall term and everything last week. But anything I can do to sort of facilitate that for DevoWorm specific projects too. I'm interested in hearing more about that or facilitating what I can. It's my brief update for today.
Yeah, I think that Lucas's project is. He gave an update in our Saturday meeting because he's been having trouble finding time to update us on things, but he's coming along on some of the work that he's been doing. So he did some bioinformatics work for the paper we just published and he's doing the studentship through OpenWorm to kind of make that more sort of, build a pipeline, make that more computationally explicit. He, did a lot of work kind of in the way that a lot of people will do work. They'll have their resources in different places. They'll kind of, do very informal things, steps in their analysis. And the idea is to build a reproducible pipeline using a Jupyter note or some sort of computational notebook, or some API tools, and building a set of commands and building a GitHub repository around that. So you can just go to the GitHub repository, follow the README, go to the notebooks, use those, and be able to execute a similar analysis. And then, of course, fit that into models of an embryo, which we have in part. And, it augments that model quite a bit. So, yeah, I think that's good. And then as far as so he's making progress, and I said, we'll have to figure out how much is sort of the right chunk for a studentship project, because you can't do everything during a studentship, but you can do get to a point where it's something you can showcase. And then that's something, also we can integrate into the things we're doing. So, it's nice to have those kind of opportunities. As for advertising DevoWorm stuff, yeah, we haven't done that in a while. We talked about this also Saturday, and there are a lot of ways you can advertise different opportunities, and so I'm going to work on that. Maybe we advertise specific projects. I know we have the Google Summer of Code projects and some of these other studentship opportunities, but also other projects that are kind of moribund right now, and I'd like to see them kind of.
When I'm done, which hopefully will be through this week, I'll show what I've done with Joko. Part of what I want Joko to be is sort of a not quite like a jobs board or a quest board, but something that can offer a space to see certain almost more like a showing projects that are being launched. Because I think that's a key thing both in this space and in RL and in other places too. I know a lot of smaller startup groups that are trying to just explain, so much of the barrier is just this explanatory, or putting things in a way that's easy to understand and understand. Okay, if I do this, I can do that, and it's a challenging thing to do. So I think there are some ways that it could be beneficial, or at least if not beneficial, not indirectly helpful too, to have it. So more about that ahead, but I'm definitely interested in yes, having the history page, which I'm updating slowly, the actual history of Google Summer of Code, but then say, okay, here's DevoWorm, here's some maybe past projects or existing and forthcoming projects, and giving it a little more of a different kind of structure. Because I noticed a lot of the continuation. There's sort of the existing DevoWorm website, and kind of what I tried to do for a while with that GitHub, the GitHub thing for RL was give a framework to what was going on, and I think I've sort of improved upon that now, and there's a sense of, okay, across many different fronts, here's different working groups and what they're doing, and sort of project associated with that, and I think that would be helpful too. So more about that. Hopefully I can finally release this through the end of this week. But I'm happy to talk more about that up ahead.
Yeah. Yeah, that's good. Maybe we need to have a reorganization a bit of things, like in terms of advertising them, getting them out. We haven't done that in a while. We've done things like build onboarding guides and other types of things. We've had the 10th anniversary reassessment, or to see where we've been. So we have that stuff kind of out there, and it's just
there's like a tremendous amount of great material already. So I think some of it's just putting that in one space too. But yeah, I look forward to that, and I'm looking forward to. I'm starting to actually. I have a sort of like movie playlist that I'm trying to watch in like my evenings to hype me up for fall. I'm actually really excited about like autumn semester and just things coming up, and this is part of that. So yeah, sometimes.
Yeah, that's great. Yeah, we should talk more about that, and try to get some things out there. So thank you, Jesse. Jesse asked Morgan, when are you in Boston this week? I know Morgan is in, I guess he's going to Boston, or he's going to Rochester, New York, and Boston. So yeah, Morgan's here, and Pakhi, of course, we talked on, I think you gave a presentation at Friday's meeting on some of your progress on the work you're doing for GSOC. Yeah, and so yeah, that looked pretty good. I think you're getting things together. I can offer you an extension for your project for maybe 2 or 3 weeks. I'll have to communicate with the people at INCF about that, but it looks like you're making some pretty good progress on your project. So yeah, I know Pakhi can't talk today, but like, did you want to put anything in the chat and mention anything about your project today? Put it in the chat. Looks like Morgan isn't sure where he's going to be this week, so it's fine. Yeah, and so Jesse and Morgan might connect this week. We'll see in person. It's always fun to get together in person. I don't know if Susan is able to present yet.
Yes, I could.
Okay, yeah, go ahead.
Okay, so this is about contraction. To produce a wave that we've been talking about, like we've talked about waves in tissue, and it's basically cell contraction or expansion. And if you do this with epithelial tissue, it's sort of two-dimensional, not exactly, but you're dealing with fewer cells deep, or at least I'm currently working with a model that's only 1 cell deep, and I know that's not true of most tissues, but anyways, you can actually physically make a tissue that's 1 epithelial cell deep, so you could test it in reality. Anyway, so here we are. They wanted to know the biological background, so the contraction of 1 cell leads to the contraction of the cells next to it. And these contractions occur in epithelial tissue and are thought to lead to cell differentiation if the cell contraction lasts long enough for the forces to get into the nucleus and rearrange the cytoskeleton of the cell and rearrange some of the nucleus. And an epithelial cell has an interesting structure. It has an actomyosin ring around its apex. So this is an interesting structure leads to interesting mechanical properties.
Yeah.
Anyways, so this is a side view, and the attachments are all along the top here. And there's actually a trijunction in between cells, a trijunction, bijunction, quadjunction, whatever is needed. And what I'm trying to model is a cell contraction in this cell. We'll make the top narrower and cause it to be more columnar. And this will affect the next cell to do something. And then that'll affect the next cell. So that's what I'm trying to model.
Right.
And here's an actual real model of an epithelium with contraction going across the top of the tissue. It says modeling epithelial tissues as active elastic sheets.
Yeah.
Reproduce contraction pulses and predict rip resistance. It's kind of an interesting paper.
So what they're doing here is they're trying to
No.
model like the effects, mechanical effects across a sheet of cells, and looking
Yes.
at right.
Right. This is across a sheet of cells, and just for simplicity's sake, I'm just going to do a row of cells.
Okay. Yeah.
For the modeling. I mean, hopefully get to this stage, but to start with just a row of cells.
Okay.
Go lay down.
Anyway, so the problem motivation, the hypothesis is cell contractions that last long enough for forces to be sustained in the nucleus for long enough to affect genetic expression. I could have probably stated that differently. And basically, this is the link between mechanical force and genetic expression.
Yeah.
So modeling goals, would like to model the force and see where it goes. I need a quantitative modeling so that you can track the force between one cell and another. So it's a simulation that models what a group of epithelial cells experiences when one of their members contracts. And the energy of this wave or contraction is thought to be a phase transition. You're actually changing the cytoskeleton of the cells from sort of cuboidal to columnar cell. And cells have a refractory period where they can contract for a while and then they can't do anything. Then they relax and can't do anything for a while because they've used up their chemical reserves. And here is the CC3D simulation that they've already done of an epithelial string of cells. So they figure they've already got this as a model. So this is the ECM or basement membrane in gray. And then the bottom of the cells is in blue, the middle part of the cells is in green, and the apical part of the cells is in red. So they make it in three parts, as three compartments, actually. So I was a bit disappointed about that, but it's true that the apical part of epithelial cells is totally different from the rest of the cell.
Yeah. So the three compartments there, what? Or the apical, the basal, and the what?
Yeah, the basement membrane is in gray, and the basal part is in blue, where it's attached to the basement membrane. Green is the middle part of the cell, and the red is the apical part of the cell. So you attach the apical part to each other, like that would be connected by a force. And they've actually got that in a model where you're disconnecting the apical part of the cells to each other.
Okay.
And the rest are, well, the blue, the basement membrane to cell, that's connected to each other. But they're not necessarily connected across.
Oh, okay. So they're
I guess
connected vertically and not horizontally, or?
The blue is connected vertically,
Okay.
and the red is connected horizontally.
Okay.
Yeah.
I see. Yeah.
And the green, not at all. That's just the middle part.
All right.
Okay. So what do we need? We need epithelial cells on a substrate. You need calcium buildup and actomyosin buildup. And then you need to add ATP, of course. So the cells will cramp at the top to make the area of the top of the cell smaller and make the cells into a columnar type cell. And this should cause a cascade of cell cramping in the epithelial layer. So to model this, to model the epithelial layer 1 cell deep, place attachment forces between the tops of the cell where the actin ring is, and model a cell cramp. And this is what I gave for resources.
Oh, yeah. Yeah. That's good.
Yeah, so especially the first one, that's so that people can see an explanation and get it for free. And the same with the last one here. I said this is more complicated. The last one reference is more complicated than is needed, but the introduction has lots of resources about the waves, the cramp waves that go through epithelial tissue. And I actually had someone ask me, so they said, is that true of all epithelial tissue? And I said, yes, it's non-muscle myosin, and it's associated with actin, and you add calcium and ATP, and you get a cramp. It's a similar mechanism to what happens in muscle, but it does move.
Right.
And they didn't realize that. And she was very into genetics, so I'm glad I was able to point that out to her.
Right.
Oops.
Yeah, myosin is just actually just in muscle, so like that's the thing that's responsible for the movement in muscle, or the right. So it just works
it's a ratcheting action.
Yeah.
And it's very, very similar to what happens in epithelial cells. And it's non-muscle myosin, but the ratcheting action of the actin and myosin is very similar, associated with calcium and ATP. So anyway, that's my slides.
Well, that's
Any questions?
Yeah, that's great. Did we have any questions from anyone, or? So yeah, if you could go back to slide 7, I just wanted to make a point about that. So that looks like a nice array, like it's this 2-dimensional array, kind of cut a cross section. So you said that the red parts were connected vertically, and the red parts were connected horizontally across that array, and then the blue ones were connected vertically up and down from the bottom to the top.
Yes.
So that's interesting, like from a network perspective, how you could model this as like, a network of forces, and how they're kind of not all going in the same direction within that cell sheet. So you have forces kind of from the substrate, forces from the neighboring cells, and how do those kind of work in a network?
Yeah, I'd be interested in working with that. Actually, I hadn't thought of that, but that's the way my tensegrity models are.
Yeah.
They're very sensitive to their attachments, both the basal and the side attachments, and the pre-strain. That seems to be what they're sensitive to. That's what they need, or something. That's what changes the graphs.
Right. Yeah.
Yeah.
Yeah, that's good. Yeah, thanks.
Okay.
So this is something that you could model in CompuCell 3D. I mean, you could do it, but they don't have. You said that they don't have like a good model of like mechanics included in the platform.
It's sort of there. He did say, well, that's almost directly related to the forces involved in cells, and you can. I have to get out my notes to see exactly what he said, but yeah, some of it is directly related to the forces involved.
Yeah.
But it's not, like I said, how many newtons is that? Like, give me hard facts, newtons. How many newtons?
Right.
And they said, well, it's sort of the forces involved. Yeah, no, I can't use this.
Yeah. It doesn't have like in the force in newtons, and then yeah, I know what you mean.
Yeah, if it's not got, well, I think you can do size properly, like micrometers.
Right.
But whether you can do nanotech with it or not, meaning when you get small enough, surface area forces matter more. Oh, okay. I'm not thinking of it again. And I'm not going to right at the moment. I have interference here with the dog. Oops, the dog is helping.
Okay.
So there are effects at the nanoscale that are not at our normal microscale. I took a nanotech course.
Right.
So
Right.
forces that are stronger at that nanoscale, and they affect cells. So you have to keep that in mind too. You're going small, so you have to watch out for that. So I don't know if the CompuCell 3D has done that, but I'm sure you could put it in your model. Like it's a
Right.
fairly flexible model. And he says it's great to try things out with and see if it's trending in the right direction. And then you can think to go to their tissue forge, which they've just developed, and do a more detailed model with more forces and more properly modeled.
Right.
But it's really great to try things out with. That's what he said.
Yeah, we
And
covered
you can
tissue
do
forge.
you can do chemical fields with CompuCell 3D,
Okay.
which is nice. That's why I said, oh, there has to be a calcium field. And
Right.
you can just put that directly into your model.
Yeah, that's good. Yeah, and of course we have these nanoscale effects when you're at a nanoscale that you don't see at the microscale, and if you're in that regime, then you have to model it. It's basically a different set of things that you're modeling. So you have, if you're going to have like something that interacts between the nanoscale and the microscale, you have to account for both types of effects. And yeah. And then we talked about Tissue Forge a while back, and that's like their new sort of where they're doing multi-modeling or multi-scale modeling. They're basically bringing together a bunch of different types of models.
Yeah, if you want to hear about Tissue Forge, they have the presentation about it on YouTube that goes with this conference. They had, there's a medical doctor who's very involved with Tissue Forge, And I don't know that I could get involved myself with my model because I'm out of time.
Yeah.
Like I should probably just go with what I have, write it up and say, here you go. And then mess around with Tissue Forge.
Yeah, I mean, it's probably a whole new frontier. So
Yeah.
it might
yeah.
take a while to learn it.
But they were very pleased that I was interested in it. So
Good. Yeah. Yeah. So yeah, I've been trying to kind of figure out a way to like incorporate this sort of CompuCell 3D, world into what we're doing, and it's been hard because it's a hard learning curve, as I understand, steep learning curve for applying it to a specific problem, and then we have to have the specific problem that we want to solve with it. So, and then as you're showing, it's not as straightforward as just taking a problem and sticking it in there.
yeah, well, you need to know what you've modeled and the fact that it's a model and not reality, and you made these shortcuts. One of them is that you made the affinity to each part of the cell so that it'll stick together, and then you have to make it so it's lesser between cells so that they don't mix. And you can't put a strong force on the top. I don't know. I'll try and do that model with them so I can get a better handle on what's going on with CompuCell 3D so that I can maybe write a bit about it in my thesis, and that would be good enough.
Yeah.
I think it'd be good enough if I had, well, I have already written something about it, but I might as well do something more comprehensive in a paragraph or two.
Yeah.
You can see my family in the background there.
Yeah.
And my son at the door. Yeah, anyway. All kinds of interesting things, and the 4 dogs. And yes. Anyway, so is it good enough?
Yeah, that's great. Yeah, thanks for sharing. This looks good and promising. Yeah.
Okay.
All right.
All right.
All right. So yeah, thank you, Susan, for that update on CompuCell 3D and how to use that, how to implement something in it, or how to pose a problem so that we can use some existing modeling platforms and kind of fit that together. So that's always a problem with modeling is you have like some problem, you make a problem, you have a hypothesis or you have a problem you want to put into a model, and you have to figure out what modeling paradigm you want to use. So we do a lot of stuff with machine learning, and of course you have to train those models. You have to not violate the assumptions of those models. And so we've been doing this with Pakhi's project too, where you have to fit the problem that you want to solve into the model, and given its constraints and its assumptions, how do you get an output that tells you something? So that's the biggest challenge of modeling is to get pick the right type of model. Maybe you don't have everything you want, so maybe you have to add things in, but that's kind of hard to do. In open source models, we can theoretically add things in, but it takes a lot of overhead. And then get an output that's that's reasonable. It can tell us something about the system we're interested in. So that's how you approach modeling, and it's not a perfect exercise, but it should lend, if you're successful, it should lend enough insights so that you can say something interesting. So, okay, so thank you for those updates and presentations. So yeah, I'm going to talk about a paper here for the last maybe 15 minutes, and it's a new paper that's come out on C. elegans. This is, again, kind of in the direction of molecular biology, and C. elegans is a good model for molecular biology because we have full genome. We know kind of the identity of every cell, and we know a lot about the organs. We know a lot about the defined mutants and so forth. And so it may seem like C. elegans is a really good system to build molecular models. So we, maybe know what certain genes do if you knock them out, if you mutate them. And we know a lot of the gene regulatory networks for certain things. We have the genes defined and so forth. But of course, there's a lot of variation there. And people have studied, the sort of the epigenomics of C. elegans extensively, although there's still a lot more to be learned. And the epigenetic effects in C. elegans aren't as great as, say, in mammals, but we still have those. So there's this new paper that came out. I think it's a bioRxiv paper. This is called Alternative Splicing Across the C. elegans Nervous System. So as you're aware, C. elegans has this complete connectome, and we know what the neural cells are, and we know what the neural circuits do. So we can identify things like a movement circuit, and we know kind of how that's activated during forward and backward movements. We have we know something about the sensory motor loops within the connectome. We know something about things like chemosensation and other types of the effects of other types of stimuli. But we also have these things that are going on with regulating the nervous system at the genetic level. And so they're exploring alternative splicing. And you'll find that in C. elegans, people produce these papers that kind of are this genome-wide or this organism-wide sort of attempt to characterize something that we know, like a concept from biology, and we're applying it in the C. elegans. So that's what they're doing here. And they're applying this concept of alternative splicing. So let me go to the abstract to kind of explain what alternative splicing is. So alternative splicing is a key mechanism that shapes neuronal transcriptomes. So what alternative splicing is when you have a gene, and a gene will often have coding regions or open reading frames. And those open reading frames will allow the transcriptional machinery to read certain parts of the gene. And so when you take your coding region of the gene and you express it, you get these transcripts that sort of get produced as mRNA. And those mRNAs then can be spliced in different ways. So you have different ways that mRNAs can be spliced, and that allows you to take a single gene or a single coding region and produce multiple isoforms of that gene. So, what you're basically doing is in the genome, you have these splicing sites identified. Different parts of the coding region will be cut out, and then you'll have these different splicings of the same gene or of the same coding region. And so these different isoforms have functional consequences. Oftentimes, different isoforms will be used in different sort of regulatory settings. Sometimes, any one gene can be expressed, and depending on the different isoforms that exist, it can be sort of regulated in a contextual manner. And this is especially important in the nervous system because you're doing a lot of things in the nervous system that require this sort of graded expression, when you need to have this sort of graded expression of things for function. So like if you're moving forward versus moving backward, maybe alternative splicings are useful there because it would be much less useful to have multiple genes that are basically duplicates but doing slightly different things. It's more efficient to have one gene for that, or having like duplicate genes but that basically don't change that much across evolution, but can be spliced in different ways and recombined in different ways. So that's basically what alternative splicings are, and so that's why they're important. So these alternative splicings help to define neuronal identity and modulate function. So you can also have alternative splicings being involved in cell identity. So when you have a certain cell type, that those alternative splicings might be different than in a related cell type that has some different function. So we can modulate function, we can define neuronal identity, and those alternative splicings are important in that. Here we present an
How do the
atlas.
splicings come about?
What was that?
How do the splicings come about again?
Uh,
How do the splicings come about?
yeah, usually if you have like a transcriptional event, you'll have your coding region that's transcribed, and then there will be some sort of post-translational mechanism that will splice those different parts. They're defined within the coding region, and there are splicing sites, and they're identified post-transcriptionally, and then those mRNAs are spliced in different ways. So you might take out a region of the code, you might take a chunk out of the coding region and combine two parts of the coding region that are maybe far apart. Or you might take out like the beginning of the coding region. And it just depends on the context. So there are these post-translational mechanisms that do this. Does that answer your question? Or?
Yeah, thank you.
Okay.
I should probably watch a YouTube video.
Yeah, that'll give you more detail. I'm just kind of glossing over it right now. So here we present an atlas. So we have an atlas of alternative splicings across the nervous system of C. elegans. Our analysis identifies novel alternative splicings in key neuronal genes. So, you can identify key genes and those have a large effect, but then the splicings give it this sort of range of function that you wouldn't get by having like duplicate genes that have like distinct functions themselves. So that's basically it. So UNC40, DCC, and SAX3 Robo are some of these key neuronal genes that they're interested in. And not all genes have the same potential for alternative splicings. Some have more of a tendency for it than others. And again, this has to do with the functional imperatives of those genes. Globally, we delineate patterns of differential alternative splicing in almost 2,000 genes. So, you can look across, your candidate genes, or you can look across the genome. And if you look across the genome, you see that there are these patterns across 2,000 genes. So alternative splicings are ubiquitous, but only, some genes are more apt to having a lot of isoforms than others. And estimate that a quarter of neuronal genes undergo differential splicing. So this is where you have this distinct functional, imperative for splicings, or where they serve specific functions. We introduce a web interface to examine splicing patterns. We explore the relationship between neuron type and splicing patterns, between splicing patterns and differential gene expression. So again, you can look at different neuronal types and look at how splicing patterns exist within the same gene, or you can look at, at gene expression that's, differential gene expression, and look at the splicing patterns that result from that differential gene expression. And I guess it's just because, you have this in different neuronal types, you have different things going on in a regulatory sense. And when you have differential gene expression, you'll see that sort of turned on and off. So it's just kind of a difference in terms of, whether it's maybe important for a neuronal type or important for function or whatever. We identify RNA features that correlate with differential alternative splicing and describe the enrichment of microexons, which exons, of course, are these regions, you have introns and exons. And so this is where where these become important. Finally, we compute a splicing regulatory network that can be used to generate hypotheses on the regulations and targets of alternative splicings in neurons. So they're working on the splicing regulatory network. So there's a lot of content in this paper. And so this kind of goes over differential alternative splicings as sort of their fundamental mechanism, elevating molecular diversity. So the idea is to create a lot of molecular diversity from, you don't want to have an infinite number of genes. And there's people still are kind of unraveling this idea of why genomes are a certain size, right? Like, why don't you have a gene for every single thing in the organism? And the answer, of course, is because it would be, very too large to fit into the nucleus. Maybe there are also energetic imperatives. And we talked a couple weeks ago about cell division and how cell division takes as long as it takes to replicate the genome. So if you have a larger genome, it takes longer to replicate the cell, to replicate the DNA, to have a cell division event. So your cell division event takes longer. So we don't want to have an infinitely sized genome. And so this is one reason maybe why we have alternative splicings, because it gives us that diversity without having huge genomes that are take a long time to replicate and to pass on to the daughter cells. And they kind of talk about alternative splicings. They occur when a pre-mRNA is processed in more than one way. So this is where different introns are removed. So you have introns and exons. The introns are those spaces in between the exons. The introns are removed, and the exons are left. And so as long as you have this pattern of introns and exons, that kind of gives you an idea of how the structure of the alternative splicings in that coding region will, it gives you sort of a combinatorial model of what's possible. And then depending on the situation, different introns are spliced out, the exons are left, and that's your alternative splicing. And then what you end up with then are these mature mRNAs with different sequences, and those different sequences, of course, have an effect in building proteins, in building amino acids, proteins, and then tissues. So you get different protein isoforms as a result. AS can also affect the stability of other features of the mRNA itself. The vast majority of human genes undergo alternative splicings, and defects in alternative splicings have been linked to various diseases. So you can have alternative splicings as this sort of mechanism for diversity, but you can also have defects in this process that lead to diseases or other types of dysfunctional aspects of the cell and the tissue. So this is a process that's regulated spatially and temporally. So you can have, alternative splicings that vary across time. You can have alternative splicings that vary across different cells. So space. So if you think about your tissue, different cells in the tissue might have different complements, different sort of stoichiometries of these isoforms. And so that may affect the phenotype. You might also have this over time. So like if you have this sort of early in the process of, if you say have like some movement, and you have a forward movement, a backward movement, maybe alternative splicings might play a role in, switching from forward to backward movement. And of course, I don't know if there are any papers that specify that, but this is just an example of how this might be useful. So in the nervous system where it's most prevalent, and there's some citations here, differential AS controls multiple aspects of neuron identity. So it controls identity. And of course, cell type identity is, there's this sort of the larger cell type identity, like you might say something, is a glutamatergic cell, or it's some sort of interneuron. And then you also have sub sort of identities where a subset, say, of interneurons do something. And so, neuron identity can become very sort of specific depending on the function that you're looking at. So differential AS controls multiple aspects of neuron identity, including global alternative splicing switches during development. So there are different switches that occur, and this is in citation 5. Isoform differences in neuron type specification. So depending on how different neurons are specified, there are differences in isoforms. And that's citation 6 and 7. Axon specification, guidance, and synaptogenesis. So the formation of synapses the formation of axons and how they're guided to other neurons. That's in citations 8 and 9. Synaptogenesis is particularly important in larval C. elegans, but also, with respect to neuronal plasticity. In terms of synaptogenesis, you do have some plasticity in the adult as well, and has been linked to several brain disorders, and this is in 1, 10, and 11. So this is stuff that's, I don't know if this is specific to C. elegans, but it does give you an idea of how alternative splicings operate in the nervous system and what they're responsible for. Alternative splicings have also been studied in cancer. Indeed, some forms of cancer are dependent on differential alternative splicings because they're not cell specific. So, you don't have like specific alternative splicings for a cell type, but you do have them involved in different processes. They can be identified by methods such as sequencing bulk cDNA. And so this is a method where you can actually look at the cDNA and get a sense of, what these alternative splicings are. So, it almost has the feel of like almost like a hammer trying to find a nail in a way. Like you have this mechanism and it's cool and you look for different things that it's involved with. And so this is why we have all these different studies on these different things. But as to what specifically specific roles of alternative splicing and where they have the greatest effect, that's still kind of an open question. But that's why they're building this atlas, so people can go back to the atlas and make connections and ask higher level questions and things like that. So they have this nice literature review. I think it's so in this paper they analyzed data generated by the CenGen project, which is an important project in C. elegans, where they're doing things with C. elegans genetics. They produced this atlas of alternative splicings for 55 single neuron types. We develop analytical tools to make the study of this easier, and they study differential alternative splicings between neuron types. So their data is shown as sort of the differential expression of the differential presence of different alternative splicings between neuron types. So if you have different neuron types, you can see the difference. So these alternative splicings are present in these cells, but not these cells. And so focusing on canonical alternative splicing events, we establish overall patterns of differential splicing. And then, of course, they developed a computational approach to extract a regulatory network for differential alternative splicings. So this kind of shows their methodology here, where they're using FACS to sort cells. So FACS is a cell sorting technique. They sort the cells by their identity. They extract the mRNA. They use these RNA-seq counts, so they sequence and align the RNAs. The RNA-seq, of course, maps RNAs to the mRNAs to the genome, to different genes, different locations in the genome, but also counts the number of transcripts. So you get this quantification that tells you how much of a certain transcript you're getting. And of course, in an alternative splicing, you're going to have it mapped into that coding region. You're just going to exclude the introns. And so this kind of shows the procedure. This figure kind of shows a comparison to previous splicing data. And so they show this in DAF2, which is a gene in C. elegans, and they show kind of the connections between these different splicing, or show the different splicing patterns and what they might look like. This is detection of a novel cassette exon in UNC40. So they show in this UNC40, they show this pattern of splicing here. And they show it, the evidence for it in the blot here, where they have the different parts of the splicing down here, and they can show them sorted by molecular weight. They show that in the gel. And then they show these exons located in the coding region here. Okay. This shows, some more molecular biology, the differential AS between neuron types. So again, you use a heat map to show the differential expression of different alternative splicings. So you have in this figure here, you have different cell types. So you have IL-1, IL-2, AVA, AVE. You see, like, of course, the intersection of the same cell as gray, and then the expression of these alternative splicings in different shades of blue. And so that's what that's just showing. They have this sort of by neurotransmitter type. They have you can identify different you can identify cell types by neurotransmitter. You can, and this is what they're showing in these colored bars down here. So neurotransmitter type, ciliated cells, either yes or no, and their modality. So you have interneurons, motor neurons, pharyngeal neurons, and sensory neurons. So you can sort all these cell types these 55 cell types by their modality, by their status of being ciliated or not, and the neurotransmitters that they express, and then look at those differences here. So it's a nice figure to look at some of this diversity. Okay, so now they get into some of these networks here. This is a splicing regulatory network. So you have the alternatively skipped exons, the putative splice factors, the covariance matrix, and then the precision matrix. They show these different, the spliceosome network, they call it the spliceosome in other papers, but basically this shows your coding region linked to different exons, and then they show this for different genes. I'm not really sure how this is structured. So, there's a lot of methodology that I'm skipping over here. But basically, they build these networks out of the spliceosome. They use some different algorithms to discover these spliceosomes and then to build these networks. And then they, validate it through different statistics. So I think that's a good overview. I know that's a pretty complex paper, and I don't know how useful it is. There's an associated atlas online, which I'm not going to go into right now, but it allows you to explore the data. And usually the atlases are at a higher level than the paper, so you can query specific genes, specific things that you want, specific sequences, and it gives you that information. Now, linking this to the phenotype, of course, they did a little bit of that in the paper with some of the different, neurotransmitter types and things like that. But it's maybe a useful tool if you're interested in the connectome and, thinking about some of the functions of cells in the connectome and, thinking about the connectome sort of as a functional entity, not just as a structural entity, which a lot of people tend to think of it. They think about the connections between cells. And of course, they're not static. They're varying all the time in terms of how they operate in a circuit or how they do things over long periods of time. So that's, again, I don't know. It's kind of hard to link a lot of molecular stuff to, the phenotype of the connectome, but that's what we're trying to do here. Okay, so Okay, if there's nothing else, I think we'll call it a day. Did we have any questions before we go? Comments?
No comments, just, I guess, thanks for the meeting, and I'm going to have to explain the meeting to Dr. Sharif because you have my name on a paper. Oh well.
Yeah. All right. Well, thanks for presenting, Susan. That was a good presentation on that.
Okay, thank you.
All right, thank you. All right, everyone. Talk to you next week.
Bye.
Bye. Thanks. All right, now I'd like to talk a little bit about morphogen gradients, and I have two papers here that sort of reassess gradient-based thinking and morphogenesis, and then a more detailed paper, mathematical paper, talking about interference in morphogen gradients. So this first paper from the International Journal of Developmental Biology, this is from 2006. This paper is called The Role and Limits of a Gradient-Based Explanation of Morphogenesis, a theoretical consideration. So, as we know, gradients are often what we use to describe morphogenesis. We have a source, which is usually some chemical, and that chemical has a gradient. And so if we think about this in terms of an embryo, or maybe like the type of epithelial sheet that we were talking about earlier, let's do an epithelial sheet just to kind of give an idea. So we have these cells in an array, two-dimensional sheet. Just for demonstration purposes, and each one of these cells is going to have a chemical source. And so let's say that there's a chemical source being expressed in this cell. And the expression of this chemical source is greatest in this cell. And the chemical, of course, will diffuse in this direction. So this arrow, this vertical component, is the expression level. And this horizontal set of arrows is the diffusion. And so our diffusion, of course, will go across the sheet of cells, but the level will decrease as you go farther away from the source. So you end up with this gradient that is basically shaped like a bell curve in space. And so you have your diffusion that decreases as you go outward, and eventually the amount diffuses to almost nothing as you go far enough out. Now, if all of our cells are expressing this at the same rate, then the gradient is sort of irrelevant because all of them sort of have their own supply of this chemical. But if not all of them are expressing it, then the gradient becomes important, and they get this chemical but at differing amounts. So that means that the effects of the chemical are going to decrease as you go towards the end of this sheet of cells. So these cells on the end are going to receive less of the chemical than the ones in the middle. Okay, now the thing is that if we replicate the sheet down here, and we get, let's do 8 cells here. Okay. And we have the same sort of situation, but this time the cells on the ends are going to express some chemical at a very high level. These are going to be our sources of our chemical A and our chemical B. And so chemical A and chemical B are going to be sort of endogenous to these cells only at the ends. And they're going to diffuse outwards. So they're going to diffuse to the left and to the right. But in this case, we're only interested in the rightward component in A and the leftward component in B. So we have A rightward and B leftward. And so these are going to continue out, and this gradient will decay as we go outwards. And as you see that they intersect at some point. So you can see that at sort of this point, they sort of intersect. You get this, and it's not quite even, but you get this effect where they're sort of, just kind of the minimal amount at this point. But what happens in this region? So in this region, you get an interference between the two chemicals. And oftentimes in chemical morphogenesis, that sets up a gradient. So you have, if we draw our bell curve tails here, you have, the decay, which decays to almost nothing, but you have this interference region, which is here. And then oftentimes in in, say, something like an embryo, you'll have a boundary that forms, a sharp boundary that gives you some sort of, often you'll have a sharp boundary. So if we have an embryo like this, we'll have two sources of a chemical. And these will be A and B, and they'll fuse towards each other. And you end up with a sharp boundary here because as they interfere, they kind of inhibit each other, and you end up with this boundary. And so this is how you get striping. This is how you get boundaries between chemical signals. And this is the basis of chemical morphogenesis, is trying to find it as the basis for a lot of the sort of pattern formation that we talk about. So of course, this is just a model of morphogenesis. It's a model of chemical morphogenesis in particular. And so just keep in mind that you have this sort of diffusion, you get overlapping diffusion, you get this interference, and that can lead to some sort of interference pattern, or it can lead to this boundary. Okay, so that's sort of the context for both of these papers. This paper here talks about the role and limits of a gradient-based explanation of morphogenesis. So this is kind of a theoretical consideration of these limits. And so the abstract reads, development of an organism is a multidimensional process leading to the generation of complex species-specific structures. So this is where you get different chemicals being expressed, different amounts of mRNA or different types of chemicals that get diffused across this phenotype, and maybe the striping patterns, which are those boundaries, or the distributions of these, what they call soluble substances, which are these morphogens, and this is a theoretical concept. So what I talked about, chemical compounds are, I could mean morphogens, and I could mean any soluble substance that can diffuse in a cellular environment. So the uneven distribution of gradients in soluble substances, morphogens, and specific receptor-ligand interactions are known to cause differential gene expression. So gradients can be evenly distributed, but they're usually unevenly distributed. So if we go back to our image here, we have sort of a bell curve in this example. And generally, we assume diffusion to be this sort of, even sort of decay, this linear decay. And so this is maybe one model, maybe one assumption that doesn't actually exist in nature. So you might have a skewed distribution here, is what I'm saying, with diffusion rates that vary sort of non-linearly. So this is what is kind of getting at with this, is that the typical model of morphogenesis, or at least this sort of standard model, maybe isn't as amenable to reality as we'd like to think. So this can actually, in cases where you have gradients in specific substances and receptor-ligand interactions, we know that causes differential gene expression. So when we have these different gradients that interfere with each other, we have these different regimes, they lead to differential gene expression between those regions. Therefore, gradients and morphogens are used as a causal explanation of developmental processes. So what I showed you in the gradients, these are causal. So when there's a gradient of a chemical, it triggers differential gene expression in these cells, and maybe in this middle region versus the end regions. And that results in differential gene expression, which varies across this region. So some gene is being targeted by this chemical signal. It's being upregulated, but it's being upregulated in a dosage-dependent manner. And so this is where you get this idea about differential gene expression. So that relationship is thought to be causal. And that's the assumption that he's talking about here. However, each attempt to describe developmental causality should take into account both the local fine organization and the global robustness of morphogenesis. So this is where we have to consider both this local example of this gradient across, say, like a sheet of epithelial cells, and this global relationship between gradients. The classical view of the role of morphogens will be critically considered and possible alternative proposed. The core idea of my proposal is that the main function of morphogenetic substances could be a context-dependent modification of cell behavior. So this is kind of what we're talking about with alternative splicings, where alternative splicings are context-dependent, and they can be modified based on where they're located in a sheet of cells, or the cell that exhibits alternative splicings. Those can be different in a sheet of cells and in a global context. So if you go from region to region, there are differences, and in terms of cell type as well. Both history and different features of morphogenetic fields create the framework for the activity of morphogens. So this is sort of a critique of the standard model of Turing morphogenesis, Turing chemical morphogenesis. So this talks a little bit about causality and explanation and boundary conditions. So the issue of the causal factors in morphogenesis and development is both fascinating and fuzzy. So we talk about causal factors maybe in an oversimplified manner, and so we have to think a little bit more deeply about these sort of causal factors and how they play a role. So he talks a lot about causality and sort of how it operates in morphogenesis. So it distinguishes between the what is and the what is the reason of. So if you look at a pattern and you say, what is a pattern? And you can identify that. And then you have to think about what is the reason of a pattern. So, is it the pattern that's the causal factor? Or is there some mechanism that's the causal factor that leads to the pattern? And so that's what we have to think about. So sometimes when we talk about these kind of relationships, we get them backwards or confused. Or worse yet, there's some sort of circular reasoning here, where we think about the mechanism, we think about the pattern, and then we think back to the mechanism, and we make this sort of, category error. I guess that's his position here. So it is clear that the boundary conditions or contexts are a necessary component for each physical event to be explained. But one tends to deny their causal role by claiming that only an efficient cause, the source of the change, is of explanatory relevance. So this is where you think about the efficient cause, which is the source of the change, and that's the only part of this that's relevant. And in fact, in this case, we have this mechanism, and it sets up a gradient, and you have this pattern. Of course, then we assume that there's some effect in the genotype, and that leads to the pattern that we see. And of course, we have to think about that very carefully, because maybe that's not really what we're dealing with. Maybe there's a more global explanation. So we can think about this, the chemical signal and the gradient as being sort of an efficient cause. On the other hand, it was emphasized that for real physical complex systems, like the Bernard convection, boundary conditions play a crucial causal role. So if we think about this at the global scale, there are boundary conditions, namely the boundary conditions that are characterized by this interference. It's just one example that actually also play a role in this pattern formation. So this mechanism may be important, but it's not the only aspect that we need to pay attention to. It could be boundary conditions of the global case. As Michael Polanyi argued, the same is true for machines. In his view, a machine cannot be explained in terms of physics and chemistry. Principle of structure controls the principles of physics and chemistry. So the structure actually controls the principles of physics and chemistry, maybe within a compartment or, at the global scale. It could be suggested that the notion of causa formalis is of great importance to biology, since it explains how a certain change can occur even in the absence of an efficient cause. So even when you don't have an efficient cause, you can have some sort of, have a change occur. And we have to think about this. When we observe the efficient cause in conjunction with the change, we think it's obvious that there's some sort of causal effect. But if the cause is not present, and you also have the same effect. Then, we don't usually consider that case, and this is the kind of counterfactual that you need in terms of looking at causality. Such situations are common in biology. If some function could not be performed, or if some substance is not available, so if our substance isn't available here, if we have this effect when the substance is available, obviously we can point to this kind of model and say, yes, this explains it. But when we don't have this kind of, when the substance is unavailable, we still get this kind of patterning, then what's the consequence? And a lot of times in biological systems, you'll get stand-ins, or you'll get other things that are involved. And sometimes that's, that can confuse our sort of causal model. And that's one of the problems in biology. You do have these shortcuts, substitutions that can be problematic from really getting at these counterfactuals. So in such cases, the boundary conditions play an undoubtedly causal role. So sometimes the boundary conditions can play a role in the absence of some sort of substance not being available, or that mechanism not being available. This is because development is a process that not only uses previously existing conditions, but also creates new conditions. So when we've talked about this in the context of systemic closure. And so systemic closure is where you have systems that are sort of closed in the sense, not necessarily energetically closed, but closed in the sense of causality. So you have these causal loops that kind of feed upon themselves. And that's really sort of an identifier of biological systems, of living systems, that they can create their own dynamics. And so this is something that, of course, throws a wrench in this model of causality, where you have a mechanism and you have an effect at the global scale. You can actually have boundary conditions which reinforce these things, and even in the absence of a clear mechanism, can produce these outcomes. Recently, the idea has become increasingly widespread that the role of causa formalis is the origin of morphological novelties, and is crucial and increasingly grown over the course of biological evolution. And it cites Newman and Mueller, 2001. So this is where they kind of talk about this a little bit more. Unfortunately, the consideration of the notion of a context is outside the scope of most recent approaches to morphogenesis. Instead, the efficient molecular factors, morphogens and morphogen-mediated interactions, so these again are these chemical factors that play a role in this kind of sort of a reductionist approach to looking at this, are regarded as crucial explanatory tools in developmental biology. This concept implies that uneven distribution of morphogens, usually in the form of a gradient, which is what we saw here, where the effects are non-uniform across space. But of course, we have, of course, different types of non-uniformity in this gradient. Lead to a stepwise symmetry breaking in embryos into its subsequent morphogenesis and differentiation. So there's this idea that there's this uneven distribution of morphogens, and this leads to these stepwise symmetry breaking events. And that's kind of what he's getting at. He's considering alternatives to this. So he talks about the history of the gradient concept, and this is in development. This is not necessarily, or this is in chemical systems. This is not necessarily the same thing as energetic gradients that we might use in machine learning, although they're somewhat related in terms of the mathematics. But so this is the focus here is on chemical gradients. So the idea of gradients in developing organisms is very old. Theodore Boveri seems to be the first who suggested that a gradient or differential concentration of substances determines the cell's fate. Later, Child developed a detailed theory of gradients. His idea was based on experimental results showing different regenerative potentialities or potencies in planarians and other lower invertebrates along the anterior-posterior axis. So this is showing this, but in sort of a very simple model in the planarian. It's where you have this sort of anatomical variation in this model. So in the embryo, you would have sort of in the anterior pole, you'd have a difference from the posterior pole. And so the gradient postulated by Child is a metabolic one and assumed to be generated by unspecific external excitation and its transmission. Therefore, the formation of the gradient was possible without any pre-existing differentiation. So you don't need differentiation to have these gradients form. And of course, this is this reinforcing mechanism that I was stressing before, that once you have differentiation, then you can get these sort of gradient effects, sometimes without the gradient actually being, I guess is the argument. But in this case, they were able to show the formation of the gradient without any pre-existing differentiation, which means there's something driving this to start off with. Child also recognized the relation between gradients and genes. If a gradient extends over more than a single cell, cells along the course represent different levels, and these differences provide a basis in difference in gene action, certainly an essential factor in differentiation. Okay. And so this idea of developmental potencies kept going throughout the 30s and 40s. They looked in 1939 at the animal-vegetal axis in sea urchin embryos. It was shown that limitations of the fate of the embryonic substratum is related to its position within the embryo in a gradual manner. So these are these sort of bell curves that I showed you. This result was interpreted in terms of gradients and animalizing and vegetalizing substances. So the differences in the early embryo in terms of these gradients between two different substances. The gradient view is closely related to the concept of positional information, which would come later by Wolpert. And the idea is that there are different positions in the embryo that have different sorts of stuff in them. So if we think about our model here, we have these, in this latest case, we have the animal pole, the vegetal pole, and we have this gradient that kind of forms where we have animal stuff here and vegetal stuff here. And, you can define animal stuff down here and vegetal stuff down here. And there's either some sort of smooth transition between this. So you either have this sort of slope like this, or you have this more discontinuous transition here. So that's what we're trying to do. We're trying to find the pattern of transition. So you can either have something that starts out here and gradually goes from being animal to gradually becoming vegetal, or you can have something that's animal down here and then vegetal down here. And there's this very sharp transition between them. And this is the type of thing we're interested in with gradients. We're interested in the slope of the gradient. And of course, the gradient is this nabla symbol. We're interested in the slope of the gradient and the shape of the gradient. So we're interested in, depending on what position we're in the embryo, we want to return sort of our gradient value. So we're in the gradient area. So if the gradient is very gradual, we want to know kind of where we are. And that allows us to do, to look at differences in position. And ultimately, we can apply calculus to this and other types of differential mathematical methods, which make this really interesting. So this is kind of where we're going with this sort of history in developmental biology. So this concept implies that the cells acquire a positional value with respect to boundaries and then interpreted in terms of a program determined by their genetic constitution, developmental history. The concept of morphogen was introduced, a class of substances causing a specific outcome of development. So the morphogen was introduced by Turing, but also refined by this concept of positional information. You have this class of substances that can be very generic. You can define it as any substance that you can think of, but it has to cause a specific outcome in development, and it has to have some positional, specificity. So this is important, also important in terms of this concept of the morphogen. The positional value is assumed to be generated by interactions of a cell with different concentrations of a morphogen. Morphogens are defined as soluble substances that diffuse. The first clear demonstration of morphogen gradients were, of course, in Drosophila. And this was in 1988, so this is pretty late in this process, but this is showing kind of the striping, this patterning mechanism in Drosophila. During the last 2 decades, this knowledge of morphogens has been expanded enormously, and this has led to this gradient-based theory. And so then, he talks about the logic and natural necessity of morphogens and gradient formation. And so, the following theoretical reasons imply the logical necessity of a morphogen-based theory. So first is, since the genomes are equivalent in all cells, which is maybe not a fair assumption, the cause of heterogeneity should be extrinsic. So if we assume that the genomes of all these cells are equivalent in our sheet, then the thing that's affecting the differential expression here is this external gradient and not variations in the genome. And I guess another way to put that is that there's no way to say, because the cells have a specific position, that there's something in the genome that codes for this gradient. The gradient is external and affects the expression of genes in a way that is kind of dose dependent. So you can see that in this example where you get this decay over space. So any one of these cells is going to have more or less the same genomic complement. There are no specific mutations, or there are no specific recombinations or splicing events that are done in a patterned manner that allow for this gradient. You get this gradient from this sort of external stimulus. And indeed, the whole idea of boundary conditions is consistent with this, because the boundary conditions then also constrain these cells externally. So that's number one. Number two is, it is known that specific receptor-ligand interactions are able to modify gene expression. So this is where these signals are transduced into the cell. And this is where we know that that can modify gene expression, as well as getting information from that external gradient or those external sources into the cell. Three, that ligands are external signals, so we can use ligands at the receptors, and those ligands qualify as sort of these morphogens. 4 is sort of the concluding point, thus these molecular ligands should be unevenly distributed in space in order to activate genes in different ways. How will this distribution be generated? So the theory of diffusible morphogens, which form a gradient, has seen more success in recent years. Mechanisms of diffusion have been subject to intensive study. Different experiments have shown that concentration-dependent actions of morphogens are important. For example, activin, which is a substance from the TGF-beta group, is able to act in a concentration-dependent manner. So we know that there are substances that do these things. It's not just a theoretical concept. So then he talks about the problems associated with morphogen-based explanations. So this is where he talks about morphogen-based explanations and then, gets into the problems. So as stressed above, the explanation of symmetry breaking exclusively by previously existing local asymmetry is not appropriate. So this is where we have these, potential mechanisms. This idea of symmetry, sequential symmetry breaking, is not appropriate, and we need to kind of think about some of the reasons why that is. And so basically he's arguing that some of these, like I said before, there's this explanatory regress that occurs because you already need some established asymmetry to achieve this diffusion model. So you need to have some sort of structure already in place for this to occur. And so this is what he's getting at with this point. Again, this has to do with causality. The discovery of highly robust character of developmental the discovery of the highly robust character of development contradicts the specific nature of molecular interactions within the cell. And so there's a lot of variability of the mechanisms of gastrulation in the development of the colonial hydroid Diamenopumila. So you have a lot of different ways that you can produce gastrulation in this model. This is actually typical of biology. So you have a lot of things going on with in terms of cell number and the size of the embryo and macroscopic mechanisms that can vary quite a bit. And so you still get normal development. It's very robust, and molecular events maybe aren't as robust. And so there's this mismatch between what we see at the phenotypic level and what we see at the molecular level that's coming into question. We also have this role of mechanical stresses. So, of course, mechanical stresses are factored into this model at all. And of course, in chemical morphogenesis, the assumption is that it's operating on its own. And so, we have a lot of mechanical stresses, as we've talked about with respect to some of Susan's work. And so this is where we get this, we can put that into we can put those factors into the model, but then it kind of changes the model. And so, you have to think about that. And so, it may be true that cell behavior is actually the target of morphogens, as opposed to sort of the molecular aspect of it. So Huang and Iber in 2000 suggested that the behavior of a cell can be described in the framework of an attraction-based model. They describe 5 different modes of cell behavior: quiescence, apoptosis, proliferation, differentiation, and movement as stable states. And so the idea is that there are these different homeostatic states that these chemical signals can actually target those cell behaviors rather than molecular circuits. And that's maybe what we're talking about. So again, we're considering different aspects of the cell. It's mechanical behavior, it's sort of phenotypic behavior, and putting that into our model as well. Okay, so basically in conclusion, from a theoretical point of view, any local explanation of morphogenesis is not appropriate. We need to consider both local and global interactions, and that's really the way this works. We also have to think about the action of any morphogen in the context of the morphogenetic behavior of cells and tissues, as well as the mechanical aspects of cells and tissues. So these interactions between mechanical and chemical influences are really sort of at the core of any model of morphogenesis, and of course chemical morphogenesis is incomplete in that respect. And so the second paper is from 2021. This is from Vetter and Iber, Roman Vetter and Dagmar Iber. And this is a preprint, so I'm not sure where it was eventually published, but the title is Interference and Morphogen Gradient Precision from Molecular Noise Data. So this is where we talk about the morphogen gradient very specifically and the role of molecular noise. So we're not talking about cell behaviors or mechanics. We're just focusing on this molecular gradient, focusing on its precision, but also talking about molecular noise. So this requires us to think about molecular noise, which is, of course, the noisiness of gene expression and the role of that in this process. So in the process I showed here with this bell curve, this gradient, or this gradient here, this animal-vegetal gradient, this is where we consider the gradient as this transition of this smooth deterministic process. But in a lot of, and of course that's really kind of key to sort of the replicability of development. So you get this general replicability in development where you get these gradients that have to be in the right place, and things have to be spatially distinct. But we also have molecular noise, and of course molecular noise is noise in gene expression, where you get different types of variation in terms of the amount of morphogen that's produced. Then that ranges over time, and there's this noise, and your process is either tolerant to the noise or not. So that's what they're getting at with here. So let's read the abstract and see what they're getting at in this paper. During development, morphogen gradients provide spatial information for tissue patterning. So of course we've talked about this. Gradients and readout mechanisms are inevitably variable, of course, yet the resulting patterns are strikingly precise, and this is what I mean by this replicability of development. These things are precise even though they're variable with respect to cell number, cell size, embryo size, and so forth. Measurement limitations currently preclude precise detection of morphogen gradients over long distances. So, we can measure morphogen gradients. It's hard to measure them over long distances. Usually they're local effects, and then they're embedded in this global system of gradients that intersect and do other things. So it's hard to really measure them over long distances and with the way we do the measurement now. Here we develop a new formalism to estimate gradient precision along the entire patterning axis for measurements close to the source. Using numerical simulations, we infer gradient variability for measuring molecular noise levels in morphogen production, decay, and diffusion. So our production, decay, and diffusion are all governed by this sort of stochastic process. We have to use that to determine, we can estimate the gradient and the extent of the gradient. So we start off with a lot and we end up with a little. This isn't a smooth bell curve. This is a lot of variation across the distribution, but also at the ends of the distribution, you have very little to work with. So noise can really dominate here. And so knowing what this diffusion looks like in the presence of noise is important. And so what they're doing is they're developing some mathematical techniques to measure, to sort of estimate this gradient for molecular noise levels and the variability in this gradient, and produce a gradient that can be predictive over long distances within the embryo. The predicted precision is much higher than previously measured. So this is actually suggesting that despite all the noise that you might have, intrinsic noise in gene expression, that this predicted precision is much higher, or at least it's much more robust. They don't really say that, but we can use this model and it's actually pretty predictive. This model is precise enough to allow even single gradients to define the central progenitor boundaries during neural tube development. So they pick the specific event in development, neural tube development, and they look at the central progenitor boundaries and they can actually predict those pretty well. Finally, we show that the patterning mechanism is optimized for precise progenitor cell numbers rather than precise boundary positions. So it's actually like, when we estimate these, we can estimate these for cell numbers or for boundary positions. Both are sort of spatial in nature, but cell numbers are less specific, explicitly spatial, meaning that maybe our positional information is more about the number of cells in an array rather than like a coordinate system. As the progenitor domain size is particularly robust to gradient alterations. So our domain sizes really can be variable depending on how the gradient varies, and so there's this. It's easy to fit this gradient to different domain sizes. We conclude that single gradients can yield the observed developmental precision, which provides new prospects for tissue engineering. So in applying this to tissue engineering, we need to have this sort of model that's flexible because our systems are going to be variable depending on what size, your tissue. If you're engineering tissues, they're going to be of shapes and sizes maybe not seen in nature. And so that's something that we need to have for these gradients. So in this paper, and I'm not going to go over a lot of it, they really get into very detailed mathematics. They basically talk about this morphogen gradient as approximated by an exponential function. So in this case, I kind of gave an example of the bell curve or of a normal distribution. In this case, they're modeling this with an exponential function, which has an amplitude C0 and a decay length of lambda. And so lambda is the decay, and this produces an exponential distribution that decays with a tail of a certain length. And the tail, of course, can be long since it's an exponential function. The length of the gradient, or lambda, specifies the distance over which the morphogen concentration decays. So this is where, we have the tail, which is down here, and that tail can be very long and it can extend outward. So the tail is kind of what we're interested in for some of, the farther out reaches of this gradient as it gets into these transition zones and interference zones. And so that's kind of what they're doing here. They're modeling this with respect to concentration decays and how this decay is exponential. The patterning domain is typically much larger than lambda, such that the concentration gradient declines substantially within. So for instance, the bicoid gradient length is a fifth of the length of the early Drosophila embryo. So this is where the bicoid gradient actually goes out to a fifth of the length of the early embryo. The DPB, or ecapentaplegic gradient in the Drosophila wing disc, is even shorter, at 0.11 of the length. So this is where we can take this lambda and divide it by the length and get these gradient sizes, these relative gradient sizes. And the relative lengths of the sonic hedgehog and bone morphogenetic protein, which is BMP and SHH gradient, decline from about 0.2 to 0.05 during mouse neural tube development. So in mouse neural tube development, sonic hedgehog and bone morphogenetic protein are actually quite small relative to the embryo size. And this is during mouse neural tube development. So this is where we have the specific structure that's being formed. So you can see that there's variation in these gradients, and they kind of serve the needs of what's going on in the embryo. So this is kind of getting into maybe the more the math of it from the other paper where he was speculating about the size and robustness of these gradients. In the developing vertebrate neural tube, positional information is provided by opposing sonic hedgehog and bone morphogenetic protein gradients. So this is where there is this interference region, and how this kind of is triangulated from these different gradients. Even if each morphogen pattern is only one half of the domain, the morphogen concentration at the center of the final domain will be about 10 to the fourth fold lower than at the source. So again, we're talking about sort of the tails of this gradient, and where it's actually quite a bit lower than the source. So this is where, how do you sense this decayed gradient over such distances, and how precise such patterning information will be. So when you have a very low level of the gradient in a certain location, how do you sense that gradient at a very low concentration, and how efficacious is it in doing something to that cell? So if we go to bacterial chemotaxis and the responses, chemotactic responses there, adaptation allows cells to sense concentration gradients spanning at least 5 orders of magnitude. So this is where, different cells are sensing concentration gradients in the environment. They're moving towards different sources, chemical sources, and so they have a really sensitive mechanism for sensing these gradients. So they get to the edge of a chemotactic gradient. So in the case of something like a bacterial cell, you might have a chemotactic gradient, which is this sort of patch of chemical with a source here. And you have bacterial cells, and they're moving towards the source. So they're moving inward. They're kind of navigating across this space. They're navigating towards the source. They're following the gradient. And they have this very specific sensory ability. So they're very sensitive to the edge of this gradient, because they need to be able to get to the edge of the gradient in order to get into this increasing concentration. Remember, they're moving across, say, a free environment. This isn't a cellular environment. This isn't where cells are communicating with other cells so much. This is where cells are picking up a signal that's in a gradient that's separate from their sort of, there aren't any bacterial tissues. I mean, they're bacterial colonies, but they're going towards this chemical gradient independently, at least nominally. I mean, they may give information to each other about the best way to get to the gradient, but that's not really what hemotactic responses are all about, at least the way we measure them. Whether similar effects are at work also in the neural tube is unclear, but adaptation of sonic hedgehog responsiveness has been noted. So they talk about the French flag model, which is of course a famous model of morphogenesis, and the readout position is defined by the concentration threshold C. By rearranging this equation 1 obtains this equation. As a result of molecular noise, the gradient length lambda and the ratio between gradient amplitude and the readout threshold differ between embryos. This difference translates into shifts in the readout positions. The overall readout position is typically calculated as the mean of the individual positions in the different embryos I, and then the positional error is typically quantified by the standard deviation. So in the presence of molecular noise, we can give this positional error. So, there's this error that can occur, especially when you have very low amounts of some morphogen. And this, you can model this using a standard deviation. So this gives you this noise factor here. Following definition, the readout position and accuracy of two centrally located progenitor domain boundaries we quantified in the mouse neural tube. Positional error of both the dorsal boundary and the ventral boundary were found to be about 1 to 3 cell diameters. So this is where we have these dorsal and ventral boundaries of different factors, NKX 6.1 and PAX 3, and there's this sort of this positional error that occurs. So it's not, it's not absolutely precise, but it is precise enough to be replicated. And you do have this 1 to 3 cell diameter error that can occur. In parallel, also the gradient variability was measured. Given the challenges of measuring morphogen gradients directly, we used a transcriptional reporter as a readout of signaling. And so close to the source, the positional error increased from a single cell diameter to about 3 cell diameters over time. So this error, of course, could be from within 1 cell to 3 cells. 3 cell diameters, which means that it could be within 1 cell to over 3 cells. In the center of the neural tube, however, the positional error was reported to increase from 3 cell diameters in early stages to more than 30 cell diameters later on. So what that means, perhaps, is that at the edges of these boundaries, of these gradients, it's more important to be precise, and in fact, that's what you see. You see more precision. You get these tolerances that are in the range of micrometers, or like 5 micrometers to 15 micrometers. In this, where it's where it needs to be less precise, we're in the center of the neural tube, where it's less critical to patterning, this positional error can be greater. And so 30 cell diameters is 150 microns. So you can have a much larger cell diameter, or you can have much larger error, where you don't necessarily need specification. Combined readouts of the imprecise sonic hedgehog and bone morphogenetic protein gradients was proposed to yield the higher precision of the central progenitor boundaries. So this is showing an example here, something in this 3D model here, where you have the dorsal side, the ventral side. You have these different factors being expressed with gradients. In particular, you have the sonic hedgehog gradient and this bone morphogenetic protein gradient, and they one starts at the dorsal side, BMP, ventral side we have sonic hedgehog, and they kind of intersect in the middle, and you get this boundary. So you get this sort of intersection between the two gradients. And so this is just showing this. And this is showing some of the distance from source. So it shows this function with noise as it decays. And then this is the exponential distribution. Then you see this interembryonic positional variability here, where you get this difference in the distance from source. So sometimes, you get a greater distance from source or a lesser distance from source, depending on this interembryonic positional variability. This results in a distribution of readout positions with an interembryonic mean and a positional error given by the standard deviation. Okay, so the rest of this paper is talking about these statistical gradient parameters for the mouse neural tube, looking at this example here and working it out. And then, of course, they talk about the technical limitations of these measurements. And so this is kind of what they're talking about here. They're talking about gradient readouting precision in the developing neural tube, and they kind of give some examples of that here. And so, a couple words on gradient variability as a result of molecular noise. So in a cellular tissue, the morphogen production, degradation, and transport rates vary from cell to cell. And that's a very important thing to remember, that even though the genome is more or less uniform, the kinetics of the cell are not. And so this variability ultimately generates the variability in the steady state morphogen gradient profiles. We can estimate this variability by simulating a simple reaction diffusion model on a continuous one-dimensional domain, where these parameter values differ randomly from segment to segment. To describe the steady state morphogen profiles, we solve the steady state reaction diffusion equation, which is here, in a one-dimensional domain that was split into two subdomains, a morphogen source and a patterning region. Okay. Then they use a Heaviside step function to ensure that the morphogen produced at rate p is only in the source, where it degrades at a linear rate d everywhere. So you have this production rate p, and you have this linear decay rate d. Morphogen transport is driven by Fickian diffusion with diffusivity capital D and zero flux boundary conditions. This gives you a concentration profile that follows hyperbolic cosines here, and this decay length, which is large d divided into small d, and that's the square root of that, which is of course equivalent or equal to lambda. Depends on the morphogen diffusivity, capital D, and the turnover rate, small d. So it's the diffusivity over the turnover rate and the square root of that. And so this is a numerical model that predicts gradient variability from molecular noise. You get the source, which are 5 cells here, and then the patterning domain, which is 20 to 200 cells down here. And you get this where you see this numerical simulation here. The coefficient of variation here, and this shows the results. So for cell variability, patterning length, and cell variability here, CBK. So I think those two papers kind of maybe improve our understanding of morphogen gradients, maybe challenge them a little bit. But definitely I think the second paper really kind of answers some of the questions of the first paper, but actually maybe raises more questions. It's a good modeling paper, and it shows a lot of quantitative context to a lot of these ideas. Thank you for paying attention, and I hope you learned something.